Welcome to lecture 3. This is the last lecture ever for this AP biology class. I know you're excited, and so am I. So the endocrine system, we're going to talk about physiology. We're going to start with the endocrine system. And it contains the glands and tissues that are going to secrete hormones that are going to travel over short distances, and they can only target certain tissues. So for instance, here's a target cell, and this is a form of signaling. It's going short distances to those other cells, whether it be a B cell, a T helper, whatever, okay? So, and then here's another example of a nerve cell releasing that to another nerve cell. So these are short distances. Then we have long distances, right, where it goes through the bloodstream, all of that, to another target cell. So hormones are ligands, meaning that they're the first signaling molecules. And they're either proteins, lipids, or they can be gases. So lipid hormones, or also called hydrophobic ligand, are lipid soluble. They can cross the membrane to bind with intracellular receptors in the cytoplasm. So for example, we've got estrogen, progesterone, and other steroids that we can use this with. So the protein hormones bind to the receptor proteins in cell membranes of target cells, kind of like a lock and key. And this causes the receptor protein to go through a conformational shape change, and that activates a secondary messenger. So you can see here's the hormone, here's the receptor protein, and once that is on there, like a lock and key, this is where our shape change occurs, and then that enzyme is activated. Signal transduction pathways. This should be a review. So we have reception proteins, and these are receptor or intracellular receptors of a cell, and it's going to detect a chemical signal. And then transduction is when a change in a protein stimulates other changes in the cell and amplifies the signal, often a secondary messenger like cyclic AMP is activated. And then we have a response. And this is any cellular activity such as activation of a gene and its transcription or activation of an organelle or a specific enzyme. Again, make sure that you pay attention. We have some must-know boxes throughout the lecture that you need to be familiar with and understand those concepts, but I'm not going to take the time to go over them because we have looked at them previously. Okay, so for instance, the G protein link receptors, it has a role in regulation of blood sugar. So the protein ligand, or epinephrine, is going to bind to that G protein receptor, and then a shape change is going to occur, and it's going to activate an enzyme that converts ATP into cyclic AMP, which is a secondary messenger. And again, this should be review, right? So I'm going to go through it pretty quickly. So cyclic AMP has the ability to activate kinases, and these kinases end up activating other kinases and phosphorylating them. And one molecule activating many leads to amplification. And then a final kinase can activate that enzyme that breaks up the stored glycogen in liver cells, and then that glycogen is broken into monomers of glucose that can be released from the cell into the bloodstream. And then we know that kinases are deactivated by a process called dephosphorylation because it removes that phosphate group. Make sure that you take a moment to review these diagrams. Go back into our previous lectures if you're not familiar with cyclic AMP. Okay, so then we have an endocrine signaling example. So the pancreas and the liver's role in blood glucose regulation is another example of negative feedback. Now, there are lots and lots of endocrine signaling examples, and again, my recommendation is that you focus on one, and I'm going to choose to focus on the liver and the pancreas here. So, when there is high blood sugar, a person consumes carbohydrates, they're broken down into sugars, and they are absorbed by the bloodstream. And as a result, the blood glucose levels are going to be high. So the excess glucose levels are going to be detected by chemoreceptors that are located inside of our pancreas, which is then going to release insulin to control those blood sugar levels. So insulin is a protein hormone, and it travels into the bloodstream to the liver and the muscles, and then binds to the receptors and causes those liver and muscle cells to uptake the sugar. Insulin causes the liver, which keep in mind is made of muscle and fat, causes that liver cell to place what is called GLUT transporters in the membrane so that the liver cells can transport that glucose into the liver cells out of the blood. Sugar is then going to be transported into these cells for storage, and that excess sugar is converted into glucagon, and stored. And the result is the blood sugar levels drop. It eliminates the initial stimulus, which was the high sugar levels in the blood. And this is a perfect example of the endocrine system using negative feedback to keep that glucose concentration at a set point. So this diagram basically shows you here. You've eaten something, that high blood sugar, it's going to cause the pancreas to release the insulin. That insulin is released to the tissues, the cells, the liver, and then that's going to cause the blood sugar to be released. So it's going to lower that blood sugar. The opposite can be said also. If you have low blood sugar, it will promote the pancreas to release glucagon, which then goes into the liver and raises the blood sugar. And this diagram is just showing you here the placement of those GLUT proteins, which cause them to go up here and bind and allow those glucose to come into the cell. So low blood sugar, we just looked at it on that diagram, but let's do it through words here. The pancreas is going to respond to low blood sugar by releasing glucagon. And glucagon is a protein hormone that travels in the blood to receptors on the liver as well as the muscle and the fat cells. And this is going to trigger these cells to break down glycogen into glucose and release that glucose into the bloodstream. And that will cause the blood sugar levels to increase, and then the stimulus will no longer be present. And again, another example of negative feedback. So disease and physiological problems can occur when a signaling pathway is altered. So for instance, diabetes mellitus. There are two types. Type 1 diabetes is due to an autoimmune disorder, and this causes the insulin-producing cells in the pancreas to be destroyed by the white blood cell. Type 2 is due to a defect in signaling in the fat, muscle, or liver cells, and that causes reduced glucose uptake. So you can see in these diagrams here, you've got type 1, there is a diminished amount of insulin, and so that glucose is not taken up into the cells. Type 2, you can see that there is an insulin resistance happening. I want to also point out here that these are the must-knows according to AP related to insulin and pancreas, etc. So make sure that you look those over. Let's talk about plants and hormones. So plants have the ability to produce hormones that can travel throughout the plant to a target tissue. For instance, one is called auxin, and it is produced in the tip, and it migrates to the dark side and then travels down the stem. It's going to bind to the receptors on that dark side, and it's going to cause for growth and division. And this just shows you an experiment that was done related to that tip, etc., and that auxin. But basically, all it's saying is the light is here, and here, this is the dark side. That auxin would be released, and it would cause that to expand and elongate so that it would be up straight again. Then we have ethylene, and it is produced by a plant cell, and it causes the ripening of fruits. And this is an example of a positive feedback loop. So ripening is basically the breaking down or the atrophy of plant cells. And when this happens, the cells release ethylene gas. And the gas increases the rate of ripening, causing even greater production and release of gas. So more ripening, more ethylene until the culmination, which is the fruit is ripe, and it ends up falling from the tree. So that is a positive feedback. So let's talk about reproduction. So when we have insufficient free energy, it is going to result in the loss of reproduction. Can't spell, sorry. Reproduction is usually seasonal, as we've talked about before, and results when free energy is available at its highest. So for instance, in the spring, that's when birds are laying eggs, etc. An example of positive feedback is childbirth. So in positive feedback, when the condition intensifies and the result causes the condition to intensify further and so on until there is a culminating event. So here, this is the reproductive cycle, and you can see that it is a positive. So for instance, here the brain stimulates the pituitary gland after being impulsed from the cervix, and that oxytocin is released, carried to the uterus. It stimulates contractions, pushes the baby towards the cervix, and then the head of the baby pushes against the cervix, and again causing more nerve impulse, and it just keeps going and going and going until the baby's born. So again, here is that information that you need to know. So let's talk about development. So timing and coordination is key. So the differentiation of cells is due to 4 key factors. The first is called the Hox genes. The second is unequal distribution of cytoplasm during cleavage. There's induction, which cells influencing other cells. And then there is apoptosis, which is programmed cell death. So number 1, the genes. So specific genes code for proteins needed for different stages of development. So genes that guide development in the embryonic stages are called the Hox genes. The homeotic genes. I got ahead of myself. Some famous homeotic genes are called the Hox genes because they guide the body segment and appendage development. So mutations in Hox genes can lead to major development abnormalities. And common Hox genes in unrelated species are going to once again indicate that common ancestor. So the timing of the expression, which is when they're on, and the silencing is when they're turned off, of the genes is critical. Gene expression can be affected by the environment, like temperature, etc. So we know, or scientists know, that temperature can actually affect the sex determination of some reptiles. And you can see, make sure that you can look here, you've got sex ratios, temperature, make sure that you can read these graphs between the turtles and the alligators, etc. And then this chart is basically telling you the same kind of thing. So how are genes expressed? How can they be altered or affected? So microRNAs are important in gene regulation. So the junk DNA, which are the non-coding regions, they code for the RNAi molecules. And they can be processed by a dicer creating the miRNAs. And then that fragment is going to bind to a group of proteins called the RISC proteins, or RNA-induced silencing complex proteins. And that miRNA RISC complex can silence the gene by causing the destruction of the mRNA before translation occurs, or it can also block the ribosome, stopping translation. And here, this is just showing you that right here. We also have alternate gene splicing. So the same gene can code for different proteins and different responses in different cells. And this shows you here, you've got the same thing starting here, depending on the splicing, you've got protein A or protein B. Two, if we have uneven distribution of cytoplasm during mitosis, it can lead to cells getting different concentrations of molecules, such as transcription factors, enzymes, etc. So this can cause the cells to develop differently. 3, we have induction, and this is the start of an event due to chemical signaling, as well as physical contact between cells. So these chemical signals or physical signals are called morphogens. And morphogens are basically any internal or external stimuli that signals differentiation and development. So you can see here, these are some examples of the mechanisms that we're talking about. And here is your, here's your must-know box. So the communication is going to occur between the germ layers during early development. And there are 3 layers. We have the endoderm, we have the ectoderm, and we have the mesoderm. And basically, this is just showing you the beginning of the layers developing, and then here in the late stages, you can see we have these three layers. So transplantation of cells in experiments verify that there is communication between the cells. So what they did was they removed the notochord, which is right here. And if you remove the notochord, that means that there's no neural tube development, because the notochord eventually turns into your vertebra or your spinal cord. So the moving of that notochord to a new location also led to neural tube development, but it was above the notochord versus where it would have been. And fourth, we have apoptosis, which is cell death. And this is a normal part of development. Apoptosis is just basically programmed cell death. So cell fragments are consumed and lysosomes end up degrading the molecules into different monomers. So some examples that you should be familiar with, for instance, are digit formation in human embryos, so where your fingers are. So this red part actually shows you where programmed cell death is, and that's how we end up with our fingers. And then we've also got apoptosis when a tadpole metamorphosizes into a frog. Okay, so some odds and ends review things that just want to make sure that you're familiar with related to the physiological side. The Bohr shift, and this is going to be the change in the affinity or binding ability of oxygen by hemoglobin due to a change in the environment. And this shows you the Bohr shift graph. When you exercise, you are going to produce more CO2 as you make more ATP in order to meet those energy demands. And that CO2 plus the water makes carbonic acid, so the blood pH is going to drop, making the blood more acidic. And when that pH drops, that hemoglobin has a lower affinity or less binding for oxygen. So then additional oxygen is going to be released if the blood was more basic. Also, we need to cover really quick the counter current exchange in thermoregulation and respiratory systems. So crossing over of some properties, such as temperature, between two flowing bodies in the opposite direction from one another. So the flowing bodies are going to be close to one another, but they are going in the opposite direction. So for the first example, we can help maintain a constant body temperature. So heat is going to move from an area of high heat to an area of low heat. And this will heat the cold blood. Okay, so here's an example of that. And just imagine here that we'll say that this dog is standing in snow. Okay, so here's snow. We're standing in snow. So we know that this ice snow area is very cold. So here we've got the blood coming from the heart down to the extremities, and you can see it's 37 down to 32, 26, 20, 15, 11, right? So it gets all the way down there. and it gives the blood to the tissues that it needs to, and then that blood is going to go back to the heart. Well, by the time it gets down here, it's at 8 degrees. That's very cold compared to the 37 that it started with. So, what happens is, through this countercurrent exchange, as the blood is traveling back to the heart, the heat is moving from that, just like diffusion, an area of high concentration to low, the heat is actually moving to the other vessel so that it is warming that before it gets back to the heart. It's not going to reach exactly what it was, but it's pretty close. Another example is fish. It helps fish oxygenate their blood through their gills. So water flow is going to be opposite compared to blood flow in the gills. And oxygen is going to move from an area of high concentration in the water to the area of low concentration in the blood. And you can see here's the water flow over the gills, and here's our oxygen levels, 100%, right? Here's the blood flow going the opposite direction. And you can see it starts at 5 and gets all the way up to 90. So that, again, is another example of that counter current exchange. And that, my friends, is the end of our final lecture. I will see you in class.